Bismillahirrahmanirrahim. I am Rabia from Department of Chemistry. Today our presentation topic is thermogravimetric analyzer. In this presentation we will see introduction, instrumentation, components, types, construction, assembling, companies, softwares, applications, benefits, costing. Introduction of thermogravimetric analyzer. A thermogravimetric analyzer is a sophisticated analytical instrument used to measure the changes in weight of a sample as a function of temperature or time in a controlled atmosphere. It is widely utilized in various fields including materials science, chemistry, pharmaceuticals, and environmental research. The TG operates on the principle of thermogravimetry, which involves subjecting a sample to a programmed temperature profile while continuously monitoring its weight changes. This technique enables the investigation of thermal stability, decomposition, phase and transition, and other properties of a wide range of materials. Now we see why we need thermogravimetric analyzer. Thermogravimetric analysis is a powerful technique for measurement of thermal stability of materials including polymers. In this method, changes in the weight of a specimen are measured while its temperature is increased. Moisture and volatile contents of a sample can be measured by TG. Now we see instrumentation of thermogravimetric analyzer. Instrument used in TGA is a thermobalance. TGA gives information only for substances which show a change in mass on heating or cooling. Here is a block diagram of TGA in which atmosphere control, sample holder, recorder, balance control, recording balance, furnace temperature programming, temperature sensor, furnace. Now we see components of instrumentation. Commercial instruments for TJ consist of a sensitive microbalance called thermobalance, a furnace, a purge gas system for providing an inert or sometimes reactive atmosphere, a computer system for instrument control, data acquisition, and data processing. A purge gas switching system is a common option for application in which purge gas must be changed during an experiment. The thermobalance. A number of different thermobalance designs available commercially are capable of providing quantitative information about samples ranging in mass from less than 1 milligram to 100 gram. The usual range of thermobalances, however, is from 1 to 100 milligram. Many of the balances can detect changes in mass as small as 0.1 microgram. Although the sample holder must be housed in the furnace, the rest of balance must be thermally isolated from the furnace. A change in sample mass causes a deflection of the beam, which interposes a light shutter between a lamp and one of two photodiodes. The resulting imbalance is the photodiode current is amplified and fed into coil E, which is situated between the poles of a permanent magnet F. The magnetic field generated by the current in the coil restores the beam to its original position. The amplified photodiode current is monitored and transformed into mass or loss information by the data processing system. In most cases, mass versus temperature data can either be plotted in real time or stored for further manipulation or display at a later time. Here is a block diagram of furnace. Thermobalance components. The balance beam is shown as A. The sample cup and holder are B, C in a counterweight. D is lamp and photodiodes. E is magnetic coil, and F is a permanent magnet. The computer data acquisition, data processing, and control system are components G, H, and I. Component J is a printer and display unit. Now we see furnace. Furnaces for TJ typically over the range from ambient temperature to 1000 degrees Celsius. Also, some can be used for temperature up to 1600 degrees Celsius. Heating rates can often be selected from 0.1 degrees Celsius per minute to 100 degrees Celsius per minute. Some units can heat as rapidly as 200 degrees Celsius per minute. Insulation and cooling of the exterior of the furnace is required to avoid heat transfer to the balance. Nitrogen or argon is usually used to purge the furnace and prevent oxidation of the sample. For some analysis, it is desirable to switch purge gas as the analysis proceeds. The purge gas was nitrogen during the first 18 minutes, while the moisture content and the percentage of volatiles were recorded. The gas was then switched to oxygen for 4 to 5 minutes. Now Material of known Curie point to sometime a melting point standard. The Curie point is the temperature at which a ferromagnetic material becomes paramagnetic. The ferromagnetic material is suspended in a magnetic field oriented such that a vertical component of magnetic force acts on a sample. The magnetic force acts as an equivalent magnetic mass on the TGA balance beam to indicate an apparent sample mass. When the sample is heated through its Curie point, the magnetic mass is lost, and thermal and balance indicates an apparent loss in mass. Temperature accuracy is better than 1 degree Celsius with most furnaces. The temperature precision is typically 60.1 degrees Celsius. Furnaces are usually cooled by a forced air. Many furnaces can be cooled from 1000 degrees Celsius to 50 degrees Celsius in less than 15 minutes. Sample holder. Samples are typically contained in sample pans made of platinum, aluminum, or ceramics such as aluminum. Platinum is most often used because of its inertness and ease of cleaning. The volumes of sample pan ranging from 40 microliter to more than 500 microliter. Ceramic pans are best for high volume, low density sample, e.g., foams, and for materials that react with platinum. Aluminum pans are inexpensive substitutes but cannot be used for temperatures exceeding 600 degrees Celsius. Auto samplers are available as attachment for most TG systems. With the majority of these units, all aspects are automated under software control. The sample pan tearing, loading, and the mass measurement, the furnace heating and cooling, and the pan unloading are totally automatic. Temperature control and data processing. The temperature recording in the thermogram is ideally the actual temperature of the sample. This temperature can, in principle, be obtained by immersing a small thermocouple directly in the sample. Such a procedure is seldom followed, however, because of possible catalytic decomposition of samples. Potential contamination of sample and mass measurement error resulting from the thermocouple lids. Because of these problems, recorded temperatures are generally measured by a small thermocouple located as close as possible to a sample container. The recorded temperatures then generally lag or let the actual sample temperature. In some cases, dual thermocouples are used in a control feedback mode. Output of thermocouple is the voltage versus temperature table stored in computer memory. The computer uses the difference between the temperature of the thermocouple of the temperature specified to adjust the voltage to the heater. In some systems, the same thermocouple behave as a heating element and the temperature sensor. With modern control system, it is possible to achieve excellent agreement between the specified temperature program and temperature of the sample. Typical run-to-run reproducibility for a particular program falls within 2 degrees Celsius throughout an instrument's entire operating range. Combined thermal instruments. Several manufacturers offer systems that provide simultaneous measurement of heat flow and mass change or of energy change for differential thermal analysis and mass change. Such instruments can not only track the loss of material or vaporization phenomena with temperature but also reveal transition associated with these processes. This combination unit can eliminate the effect of changes in sample size, homogeneity, and geometry. Many TG systems produce the derivative of the thermogram as well as the thermogram itself. Such derivative plots are not true differential thermograms or produced in differential thermal analysis, but they provide similar qualitative information. They are often called single differential thermal analysis. TGMS and TGFTIR. TGMS and TGFTIR is used to determine the loss in mass at particular temperature, but TG cannot identify the species responsible. To obtain this type of information, the output of a thermogravimetric analyzer is often connected to a Fourier transfer system or a mass spectrometer. So, several instrument companies offer devices to interface the TG unit to a spectrometer. Some even claim true integration of the software and hardware of the TGMS and TGFTR systems. High resolution TG. High resolution TG. In high resolution TG, the sample heating rate fluctuates so that the sample is heated more rapidly during periods of constant mass than during periods when mass changes occur. This allows higher resolution to be obtained during the interesting periods and reduces the time of inactivity. Types. There are three types of thermogravimetric analyzers: suspension system, top loading system, horizontal system. First of all, we will see a suspension system. A sample is suspended from the analyzer. Highly sensitive thermogravimetry, wide range of measurable sample volumes, simultaneous measurement of TGA and TJDSC is not possible. Top loading system. A sample is placed above the analyzer. Direct measurement of sample temperatures are possible. Can be applied as simultaneous measuring devices of TGDTA and TGDSC. Horizontal system. A sample is placed at the edge of the analyzer. Now we see construction of TGA. When it comes to the construction and assembly of TGA part, it generally involves the following components: sample holder, thermobalance, furnace, gas flow system, temperature controller, and data acquisition system. Sample holder. The sample holder is a small container or crucible where the sample is placed. It should be made of a material that is inert to the sample and can withstand high temperatures. Common material used for sample holder include alumina, platinum, or quartz. Thermobalance. The thermobalance is the main instrument used in TGA. It consists of a sensitive balance that can measure the weight of the sample holder and sample accurately. The thermobalance is equipped with a heating system to control the temperature of the sample. Furnace. The furnace provides the necessary heat to raise the temperature of a sample during the analysis. It is typically an electric resistance furnace that can reach high temperatures and has precise temperature control. The furnace is integrated with a thermobalance to ensure accurate temperature measurements. Gas flow system. TG experiments are often conducted under controlled atmosphere to prevent sample oxidation or to study the gas evolution during heating. A gas flow system is used to introduce a specific gas into the TGA chamber. The gas flow can be regulated to create different atmospheres or purge unwanted gases. Now we see temperature controller. A temperature controller is used to set and control the desired temperature profile for a TG experiment. It ensures that the temperature changes follow a specific program such as linear heating or stepwise temperature increments. Data acquisition system. A data acquisition system is used to collect and record the weight measurement from the thermobalance. It typically includes software data logs for real-time data visualization and analysis. Now we see assembling of TGA parts. Assembling a thermogravimetric analyzer typically involves several components. Here's a general guideline. Familiarize yourself with components. Review the components of your TGA system and ensure you have all necessary parts. Typical components include the sample holder, balance, furnace, gas flow system, temperature controller, and data acquisition system. Prepare the furnace. Install the furnace assembly according to the manufacturer's instruction. This usually involves placing the heating elements, thermocouples, and insulation material in correct position within the furnace chamber. Install the balance. Mount the balance onto TGA system ensuring it is securely attached. The balance is responsible for measuring the mass of the sample throughout the analysis. Set up the gas flow system. Connect the gas flow system including gas cylinder, regulator, valves and tubing according to the manufacturer's instruction. This system controls the atmosphere surrounding the sample during the analysis. Connect the temperature controller. Depending on the design of your TJ, the temperature controller may be separate unit or integrated into the main system. Connect the temperature controller to the furnace and follow the manufacturer's instruction for wiring and configuration. Connect the data acquisition system. If your TGA requires a separate data acquisition system, connect it to the appropriate port of the analyzer. This system records and analyzes the data generated during the TGA experiment. Mount the sample holder. Attach the sample holder to the balance or loading mechanism. The sample holder should be compatible with your TGA system and allow for easy insertion and removal of the sample. Verify connections and functionality. Double check all the connections ensuring that cables, tubing and wiring are properly connected and secure. Follow the manufacturer's instruction to verify that all components are functioning correctly before proceeding with any experiments. Now we see the company which made TGA. Metal Tornado instruments include Nurses Instruments North America, Hidden Isokema, PAX, Nevis Instruments, Prisca Quanta V Corporation. Now we see software used in TGA. There are several software programs commonly used in conjunction with thermogravimetric analyzer. These software programs are designed to control the instrument, collect data, and perform data analysis. Here are some examples of software used in TGS. TA instrument universal analysis. This software is commonly used with TA instrument, TG instrument. It provides comprehensive data analysis capabilities, including baseline correction, peak identification, and thermal analysis calculations. NetGIS Proteus. Proteus software is used with NetGIS TGS and offers advanced features for data acquisition, evaluation, and reporting. It includes tools for kinetic analysis, curve fitting, and peak deconvolution. Perkin Elmer Paris software. Paris software is used with Perkin Elmer's TG instrument. It enables data acquisition, visualization. The analysis allows user to generate thermogram, perform weight loss calculations, and analyze thermal events. Metal Toledo Star E software. Star E software is used with Metal Toledo TGA instrument. It enables control of the instrument data acquisition and advanced data analysis, including differential thermogravimetric and derivative thermogravimetric calculations. Shimadzu TA software. The TA 60-watt software is designed for use the Shimadzu TGA instrument. It provides control over the instrument data acquisition and analysis functions such as peak analysis and thermal decomposition calculations. These are just a few examples. Different TGA manufacturers may offer with their proprietary software solution. It's important to note the software availability and feature may vary depending on the specific TGA model and manufacturer. Now we see applications of TGA. Determination of stability. TGA is commonly used to assess the thermal stability of material. It can measure weight loss or gain during heating or cooling processes, allowing researchers to identify the onset rate and extent of decomposition, oxidation, or volatilization of a sample. This information is crucial for understanding the performance and durability of materials under different temperature conditions. Now we see characterization of polymers. TG is extensively used in the polymer industry to analyze the thermal property of polymer. It can determine the glass transition temperature, degradation temperature, and the thermal stability of polymers. This information helps in designing and optimizing polymer formulations for various applications such as plastic, adhesives, coatings, and fibers. Evolution of catalyst. TGA is employed in catalysis research to evaluate the stability and activity of catalyst. It can measure weight changes during catalytic reactions, enabling researchers to analyze catalyst deactivation, carbon decomposition, or catalyst regeneration. This knowledge aids in the development and optimization of catalyst material for the chemical processes. Quantitative analysis. TJ can provide quantitative information on the composition and purity of material based on weight changes. Thermodynamic analysis. TJ can be used to determine thermodynamic parameters such as activation energy, enthalpy, and entropy of reaction or phase transitions. Complementary technique. TGA can coupled with other analytical techniques such as differential scanning, calorimetry, evolved gas analysis, or spectrometry to obtain more comprehensive information about sample properties. Analysis of pharmaceutical formation. TGA is used in the pharmaceutical industry to study drug stability, compatibility, and formulation development. It can assess the decomposition kinetic of drug compound, determine the moisture content, and evaluate the compatibility between drug substances and excipients. TGA helps in ensuring the quality and stability of pharmaceutical products. Environmental analysis. TJ finds application in environmental science for the analysis of waste material pollutants and environmental samples. It can determine the thermal behavior of waste material during incineration or pyrolysis processes. TJ is also used to study the thermal decomposition of volatile organic compounds or hazardous substances, aiding in environmental monitoring and remediation efforts. Benefits of TGA. High sensitivity. It can provide high sensitivity. TGA can detect very small changes in sample weight, making it highly sensitive to subtle thermal events or reactions. Versatility. TGA can be used with a wide range of sample types, including solid, liquid, powders, and gases. It offers flexibility in terms of sample size and composition. Real-time monitoring. TGA provides real-time data or weight changes, allow researchers to observe and analyze sample behavior under controlled temperature conditions. Now we see costing. Balance. A high precision balance is a crucial instrument for TGA measurement. The cost of a suitable balance can range from a few $100s to several $1,000s. Furnace. The cost of the furnace, which is responsible for heating the sample, can vary depending on the type and specification. It ranges from a few $100s for basic models to a few $1,000s for advanced and programmable furnaces. Gas flow control system. TGA instruments often require precise control of gas flow during measurement. The cost of a gas flow control system can range from a few $100s to a couple of $1,000s, depending on the complexity and accuracy required. Sample pan. Disposable or reusable sample pans are used to hold the samples during TGA measurements. The cost of sample pans is relatively low and can range from a few cents to a few dollars per pan depending on the material and design. Software. TGA instruments usually come with software for data acquisition, analysis, and instrument control. The cost of the software may be included in overall price of the TGA or offered as an optional add-on. Calibration. Restore defaults. Balance calibration in a self-contained routine that prompts weight calibration the user to empty the balance pan, zero the balance, the place and calibration weight that is supplied with the instrument in the sample pan. Furnace calibration. Furnace calibration is a 9-point calibration that has limit defined by the user. All you have to do is follow the prompts, input the high and low range temperature limits and depress the start calibration button. The furnace calibration is a self-contained routine that completes in approximately 50 minutes. Temperature calibration utilize Curie point reference material. Curie point reference materials are materials that upon heating lose their affinity for magnetism. The exact Curie point is a point where the material no longer exhibits magnetic properties. This point is located at the end set of the weight loss. Below is an example of a nickel Curie point calculation, a TGA 8000 using the onset calculation at the end of the weight loss. Now we see working of TGA. Working process. A thermogravimetric analyzer is a sophisticated instrument used to analyze the thermal stability and composition of material by measuring change in their weight as a function of temperature or time. The working process of a TGA involves several key components and steps. Here's a general overview. Sample preparation. The material of sample hold. The sample loading. The sample pan is carefully loaded onto the TGA instrument. It is essential to handle the sample pan with care to avoid any contamination or disturbances. Initial weighting. The TGA system measures the initial weight of the sample pan and the material using a highly sensitive balance. This weight is considered with the baseline or reference weight for subsequent measurements. Temperature control. The TGA system provides precise temperature control over a wide range. It typically involves a furnace or heating element that can heat the sample to temperatures as high as 1000 degrees Celsius or more, depending on the instrument specifications. Data recording. The TJ records the weight data obtained during the analysis and correlates it with the corresponding temperature or time. This data is usually displayed in real time on a computer monitor or saved for further analysis. Data analysis. After a TG analysis is complete, the recorded data can be analyzed during dedicated software. This analysis may involve determining the sample's decomposition, temperature, reaction kinetics, thermal stability, moisture content, volatile components, and other relevant information based on the weight changes observed during the experiment. Data analysis. After TG analysis is complete, the recorded data can be analyzed to use dedicated software. This analysis may involve determining the sample's decomposition, temperature, reaction kinetics, thermal stability, moisture content, volatile components, and other relevant information based on the weight changes observed during the experiment. Thank you. This is all from my side.